Good morning, everyone. So I'm Will Pelton, co-founder and CEO of Phytoform Labs. And yeah, today I'm going to talk to you about how unlocking the DNA of Christmas trees could potentially improve the crops that we use today. So I'm going to start off with a question. What organism has the largest, the most complex genome? I think many of us could be forgiven thinking it's us. We're pretty complex beings. And indeed, we have 3 billion letters of DNA code in every cell in our body. That's pretty big. Compared to a bacteria or a fungus, which is just a few million letters of DNA code, it's decent. You may be surprised to learn that until recently, the record holder for biggest genome was the Norway spruce, which is commonly a Christmas tree, or used as a Christmas tree across Europe, with 20 billion letters of DNA code. That's almost 7 times more than a human. And even more astoundingly, this record was in fact smashed by the Japanese canopy plant. It's a tiny ornamental, but yet it contains 150 billion letters of DNA code. So that's a whopping 50 times bigger than the human genome. And when scientists discovered this, they assumed that the reason it was so big is because basically the plant had just copy and pasted large chunks of DNA. But actually, when they sequenced it, they found it was incredibly diverse. So there were literally millions of new genes that they'd never seen before. And just for some size context, if you unravel the DNA from a single human cell and stretch it out, it will be 2 meters long. So roughly the size of a tall human. If you unravel a Christmas tree's DNA, it'll be 13 meters long, so about the height of a brachiosaur, the biggest dinosaur to have ever lived. Whereas if you unravel the Japanese canopy plant's DNA, it's 100 meters long, so the size of Big Ben. So absolutely huge when compared to the human genome. And yet, if you look at humans, we're an incredibly diverse species, right? Lots of different shapes, sizes, etc. So imagine what is contained within those 150 billion letters of DNA. And of course, plants are a diverse group of organisms, right? There's 350,000 known species of plants, and we use them for a wide range of things. We use them for food, obviously, for our $2.4 trillion agricultural industry. We use them for materials, wood. Not great examples in here, but wood for construction, fibers for fabrics, etc. And of course, I think interestingly, medicine. So many of our drugs are derived from plants, and although many of them are synthesized chemically and artificially now, actually quite a few of them are still directly derived from plants because we simply cannot copy the complexity of those molecules. And just to emphasize how important genetic diversity has been, on the left, you can see the original corn or maize plant. I'm honestly a bit surprised that humans started eating it because it looks pretty rubbish, but we did. And using natural diversity from the world, we've managed to create this absolute behemoth of a corn cob that now supports an incredible number of calories for our society today. And what I find amazing is this was blind, right? We had this was 3,000 years of work, and we had no concept of genetics or inheritance, simply trial and error. More recently, with the Green Revolution post-Second World War, we've seen incredible gains in yields across the world. A lot of this is down to machinery and chemistry, like fertilizers, herbicides, etc. But diversity still has had a huge part to play. So one single trait, which shrank wheat from sort of head height down to waist height, that trait alone, or characteristic alone, helped increase yields by 30%. So still having huge benefits to the industry. So how have we been accessing this diversity? Well, the most, original technique was conventional breeding. So basically you get 2 plants, 2 parent plants, you cross them, you get offspring, and you choose the best offspring to then iterate. We've been doing that for 10,000 years, right? That's what supported the birth of civilization, arguably. Of course, this means we can access existing diversity, and this technique is widely accepted. As I said, 10,000 years, we've been eating these crops. The issues are, it takes an incredible amount of time to do this. Literally decades to create new varieties of crop. And as a result, it's fairly expensive, and of course, it's not really scalable, right? It takes a lot of effort to develop these new crop varieties. In more modern times, we started using biotechnologies like genetic modification, perhaps a little bit controversial, and effectively we transferred DNA from one species into another, and not necessarily a plant species, often it could be a bacteria. And the benefit of this is we can access completely new diversity, right, outside of plants. But again, the issues, quite a lengthy process and a very expensive process. And that's actually due more to the regulatory hurdles that we put in place. So at Phytoform, we're trying to change this sort of, we're trying to create a shift in the time and expense you put into developing these kind of crops into a timeline that's relevant for today. So we've developed a technology, an AI-based technology called Creative, and one of our investors has described it as the ChatGPT of plant DNA. And so rather like ChatGPT, which obviously uses huge amounts of text to learn from, we also input text, but our text is in the form of genomic data. Unlike text developed by humans, our data has very little grammar. There's no capital letters, there's no spaces, there's no full stops. So for humans, it is really, really hard to understand. But of course, for large language models, similar to ChatGPT, it's great. And so we feed in large amounts of this genetic data, which of course we have quite a lot of. So sequencing technologies have exploded and provided us with this data. It's just been really hard to utilize. So our creative technology can firstly derive meaning from DNA sequences. So, it can see a DNA sequence and understand what effect that has on the plant, its development, its disease resistance, whatever. Not only that, it can then also suggest changes we can make to that plant's DNA in order to affect a specific outcome. And I think what's the real game changer about this technology is that we can do this more accurately and more specifically than ever before. So we can do it down to the single letter of DNA code. And of course, once we've found a suggestion that would have an effect, we of course need to be able to implement it into a commercially viable crop as well, so that we can get it out there as quickly as possible. And as I said before, time is running out for this, right? Last year, we lost $6 billion worth of crops to drought alone. So these changes are happening quickly. So many of you have probably heard of CRISPR-based genome editing. It's this amazing tool that was discovered about 10 years ago now, so it's not that new. And basically, it's a pair of DNA scissors. So it's a pair of scissors that cuts DNA. But what's incredible about it is that it can do so in a targeted way. So you can target it to the single letter of DNA code that you want to edit. It's an incredible tool. And that's exactly what our technology leverages. We use this tool, and as a result, it means that we can introduce a new change and edit into a crop 13 times faster than conventional breeding. So we measure our timelines in months rather than like decades. And it also means it's 230 times more cost effective than GMOs, which cost hundreds of millions of dollars to commercialize. And I guess the other important thing is it's not considered a GMO, because we're not introducing DNA from different species, bacteria, yeast, or anything like that. We're literally just using what's already in the plant. And so we're actually using this technology at the moment to transform the industry. So we are releasing a potato and tomato towards the end of this year in the US. We have edited those crops to reduce the amount of waste they produce throughout the supply chain. And as a result, of course, we hit our first sort of focus, which is decreasing emissions. Because there's less tomato and potato wasted, it means that there's less fertilizers and less fuel used to produce them. The sort of second focus for us is on climate resilience. And this is something that our partners are obviously increasingly more concerned about, particularly in the Western world, like in states such as California, which have been really badly affected by drought and heat. We're looking at how we can make these crops more robust so that we can effectively climate-proof these crops in the future, and we can do it now rather than wait 10, 20 years and continue to play catch-up with our changing climate. And then lastly, I think what is interesting is we can also help to improve the nutrition of crops. Because we can implement these changes so quickly, we can start to incorporate more natural genetic diversity from further afield, so we can enhance the nutrition of our crop varieties. And then finally, just something to sort of leave you to think about. Obviously, we want to maintain this access to genetic diversity. It is really, really important. So I think the first thing to think about is preserving what already exists. I'm not talking about within our fields. I'm talking about globally. So in the rainforest, they're huge centers for diversity. If you think a Christmas tree, that's got 20 billion base pairs of DNA. Can you imagine what opportunities are hidden in that DNA? So every time we cut down a square foot of a forest, we're losing hundreds of thousands of potential solutions. We also need to invest or continue to invest in what already exists in our crops. So continue to conserve the genetic diversity of our crops. We need more public spending on this because we've seen continual cuts to agricultural research spending across the Western world. I think finally we need to rethink our perception of biotechnology in our food system. It can be controversial, but I think considering the pace of climate change, we really need to think about how we can use these tools to improve our food system. And this is topical because yesterday the European Union released a set of proposals for how they can now regulate genome-edited crops in a different way to genetically modified crops. And in fact, they would be exempt from a lot of the regulation across Europe. That's it. Thank you very much.